Now remember, we're in the middle of talking about genetic recombination, which is how bacteria exchange genes between each other and possibly gain advantageous genetic information. There are three mechanisms for genetic recombination in bacteria. We've covered one, transformation, wherein bacteria take up random naked pieces of DNA from the environment, and these could be from any source: human, fungal, animal, plant, or other bacteria. The second means of genetic recombination is transduction. Transduction is when a bacteriophage, a viral particle that infects bacteria, accidentally incorporates part of the host cell chromosome into a viral particle. When that viral particle is released, it incorporates the original host cell's DNA into the new bacterial cell. Here is a picture of that generalized transduction process. Here you have the first bacteriophage that has the bacteriophage deadly DNA marked here in red that it injects into the first bacterial cell. When that bacterial cell is then turned into a bacteriophage factory, it will make lots of bacteriophage particles, and those bacteriophage particles will almost always contain the bacteriophage DNA inside the particles, as they should. But occasionally, an accident happens. This is unintentional, and the bacteriophage particles will accidentally incorporate part of the bacterial cell's host cell chromosome into the viral particle. When these bacteriophage are released and escape, they go to infect other cells. If the bacteriophage that contains the first cell's host cell chromosome attaches to a second bacterial cell, it will inject part of that host cell chromosome into that bacterial cell, which could then become incorporated as part of that host cell's chromosome. This would not result in lysis or death of this bacterial cell, because instead of getting the bacteriophage DNA, which would kill the cell, it just gets bacterial cell DNA, which could end up proving helpful. Unlike transformation that we discussed in the last section, where the original source of information could be from any source or even a synthetic source in the case of genetic engineering, transduction has to be from the bacteriophage infecting these two cells. As we talked about before, bacteriophage have a very specific host range, and so they will only infect cells that are related. So this DNA exchange for transduction only occurs between related cells, or cells of the same species. The third and final means of genetic recombination, or genetic exchange from one cell to another, is bacterial conjugation. And bacterial conjugation is when one cell shares DNA with another cell. This is an intentional process on both of the cell's parts. And the only time that this would occur is between related cells that want to help each other. Normally, different bacterial species are competitors, and they don't help each other by providing each other with advantageous genes. So this is usually done between cells of the same species. Conjugation requires a structure known as a pilus. Pili look like these structures here. Sometimes there's just one or a few per cell. Occasionally, there can be many. Your book shows it as this very big bridge between the cells, but in fact, they're always just like these little hair-like structures that connect the two cells. So you can consider this a blown up version. But what happens in this case is that the donor cell has the plasmid or piece of DNA that allows it to form these pili structures, and the recipient cell does not. The recipient cell has its chromosome that it needs for survival and that all cells have, but it does not have this particular plasmid. And so as a result, it does not have the pili. The way conjugation works is the donor cell makes a copy of the plasmid, and it sends a copy of that plasmid through the pilus bridge. This is a bridge made of proteins, and that allows the DNA to transfer from one cell to another. When the recipient cell receives the donor plasmid, it now has the capacity to form pili and give copies of this plasmid to other cells. This plasmid is referred to as a F plasmid or fertility factor plasmid. And sometimes this pilus is referred to as a sex pilus. But I want to be extremely clear here. This is not sexual reproduction in any way at all. It's my personal opinion that the only reason why they refer to this as a fertility plasmid and as a sex pilus is to get your attention as students reading the chapter, because this has absolutely nothing to do with sexual reproduction. All that's happening in this case is the donor bacterial cell is contributing a copy of a portion of its DNA to another cell. There is no meiosis or fertilization. There's no sperm or egg here. So this is definitely not sexual reproduction. These different means of genetic recombination, again, allow bacteria to exchange DNA that can be advantageous. Among the most important of these is DNA involved in antibiotic resistance. DNA involved in antibiotic resistance has allowed bacteria to exchange antibiotic resistance genes, or R genes, from one cell to another, providing other species with the ability to resist antibiotics. This is one of the more likely reasons why we see such a rise in antibiotic resistance in many different species in the last few decades in the world. All a result of transformation, transduction, and conjugation.